The first drug to hit $1 billion in annual revenue wasn't a cancer drug. It wasn't Viagra or an antidepressant. It was an ulcer medicine, one that you can buy over the counter as a heartburn treatment today. But here's where it gets a little weird. The drug that dethroned it as the best-selling drug was a really similar ulcer medicine. Same thing with the drug that took the top spot from that one. It was another ulcer medicine. 3 different drugs that reduce stomach acid secretion were the top-selling drug for over a decade. So for this video, I wanted to understand, why were stomach acid medications so profitable? We'll start by learning how the first billion-dollar drugs were developed, then we'll look at what factors made these drugs so commercially successful, and finally, why this story still matters to the pharmaceutical industry today. But first, let's start with the obvious. You do not want a peptic ulcer. These are sores or lesions on the inside of your stomach or small intestine. The term peptic ulcer includes gastric ulcers in the stomach and the more common duodenal ulcers in the upper part of your small intestine. On the day-to-day, peptic ulcers hurt after you eat food, you feel bloated and nauseous, and it sometimes leads to vomiting or difficulty swallowing. Now, one of the challenges for healing peptic ulcers is the stomach's own acid. The first layer of stomach lining has features that let it withstand all that acid, but once the layers underneath it have been exposed, gastric acid can keep eating away at that ulcer, making it hard to heal on its own. So for the 19th and early 20th centuries, treatment focused on modifying diet, since doctors knew that food stimulated acid secretion. Or they'd try to neutralize the acid with antacid tablets. But it takes a lot of Tums to buffer that acid, and the relief is relatively short-lived. And in severe cases when nothing else worked, surgeons would remove that part of the stomach, then sew the remaining stomach to the intestine. Or they'd cut the vagus nerve. This is the nerve that connects the brain to the gut and triggers acid secretion through the release of a neurotransmitter called acetylcholine. So a better pharmaceutical option for reducing acid secretion wouldn't just help ulcers heal, they might prevent some of those surgeries too. Our story starts in the early 1960s with Sir James Black. We met him back in the beta blockers video since he led the team that invented propranolol, but he also popularized a new model of drug development called rational drug design. See, before Black, drug discovery involved a lot of screening. Chemists would generate tens of thousands of compounds, then they'd check to see if any of them were safe and effective as a drug. Maybe they'd get inspiration from a medicinal plant, but it was a lot of guessing and checking. Rational drug design flips the order. The team starts with a biological target in mind, like a receptor, and looks for or designs a chemical that could bind to that receptor and elicit the physiology they want. And when it came to receptor-based drugs, scientists imagined drugs could be 2 things. They could be agonists, meaning they bind to and stimulate a receptor, or antagonists, meaning they bind to the receptor but they don't elicit a response. Those antagonists then prevent the body's own molecules from binding to them. Now, these days we know there are such things as partial and inverse agonists, but this sets us up for Black's story in the 60s. See, back then, one of the leading targets for a rationally designed ulcer medicine was gastrin, a relatively small hormone that stimulates the secretion of stomach acid after someone eats food. But in 1964, a team of Swedish scientists made the case that gastrin might also lead to gastric acid secretion by activating an enzyme called histidine decarboxylase, which converts histidine in the stomach into a molecule called histamine. You're probably familiar with histamine for its role in allergic reactions, but histamine leads to all kinds of things around the body. Black had seen histamine cause acid secretion in the stomach before, so regardless of gastrin's role, his working hypothesis was that stimulating histamine receptors led to acid secretion. And if histamine caused stomach acid, then an antihistamine should reduce stomach acid. But that's when he remembered something weird. Antihistamine drugs existed at this point, and back in the 50s, he'd seen a colleague give an antihistamine to rats and it didn't reduce gastric acid secretion. And that was unexpected. How could a histamine antagonist block the histamine receptors that cause allergy symptoms, but not the histamine receptors that led to stomach acid secretion? Well, Black had seen something like this before. His background with beta blockers led him to validate the existence of 2 types of adrenergic receptors, alpha and beta. Adrenergic meaning receptors for adrenaline. So maybe there were alpha and beta histamine receptors too. One led to allergy symptoms, while the other led to acid secretion. That was interesting, but Black had a job with a chemical company already. And according to his Nobel Prize essay, it sounds like he kind of felt pigeonholed as the beta blocker guy at that job, so he left and decided to focus on histamine for the next part of his career. He would test if there was a second type of histamine receptor, what he called the H2 receptor. Then, using rational drug design, he would find an H2 antagonist drug that would block acid secretion. By coincidence, one of his colleagues at his old job happened to become the research director at a pharma company called SmithKline & French Labs. So in 1964, SKF hired Black, plus a few other scientists, explicitly to find an H2 receptor antagonist. But before they could find a drug, SKF would need to prove that the second type of histamine receptor even existed. Chemically, histamine has this little pentagon-shaped ring called imidazole, along with an amine-tipped carbon tail. The SKF scientists started by modifying the central imidazole ring, then they tested whether it elicited allergy-like symptoms, acid secretion, or both. And in 1966, the SKF chemists synthesized 4-methylhistamine. It stimulated gastric acid secretion in guinea pigs without stimulating allergy symptoms. And that meant it was an agonist for the H2 receptor, but not the H1 receptor. Success. They'd confirmed that H2 receptors existed. Even better though, later that year, another research group published more evidence that H1 and H2 receptors were separate things. The next step was to find an antagonist for the receptor, but easier said than done. During the beta blocker project, they'd started with adrenaline and made variations of it to get agonists and eventually antagonists for its receptor. So they decided to apply the same playbook to histamine. And it worked at first when it yielded their agonist 4-methylhistamine, but it took them a long time to land on their antagonist. As of 1968, they'd made around 200 new compounds, but just couldn't get a good H2 blocker. Finally, after years of tweaking the structure and coming up empty-handed, a young scientist named Michael Parsons came to the company and noticed something. Black's testing procedure for detecting antihistamine activity used a low concentration of histamine, and thus wasn't picking up the changes caused by the drug. By increasing the amount of histamine they used in the test, the effects would be more pronounced. So they retested their previous compounds with the new test, and a few of them turned out to have some degree of antagonism. Now they were getting somewhere. And it turned out that one of their earlier compounds, a histamine analog ending in a guanidine group, acted as a weak antagonist according to this new test. This became known as guanyl histamine. The scientists then noticed that longer side chains tended to be more potent H2 blockers than shorter chains. This led to the formation of burimamide in 1970, which was a true H2 antagonist. But during human trials, they learned it wasn't very bioavailable when taken by mouth, so it wouldn't make for a good oral tablet. But it was a signal that they were going in the right direction. They replaced part of the side chain with sulfur and added methyl to the ring to get the more bioavailable metiamide, and it was much more potent. Clinical trials starting in 1973 looked promising at first. It kept stomach acid suppressed long enough for ulcers to heal. But it also caused a depression of white blood cells in the bone marrow called agranulocytosis, so SmithKline & French had to abandon it. The scientists thought the side effects might be due to the thiourea group at the end of the tail, so in 1972 they created cimetidine, replacing the thiourea group with cyanoguanidine. Trials started in 1973, and cimetidine showed that it could tamp down stomach acid enough for peptic ulcers to heal in the short term. This was the drug they'd been looking for. Cimetidine got approval in the UK in 1976, and FDA approval for the treatment of peptic ulcers in August 1977. Since it was an antagonist called cimetidine, SKF gave it the brand name Tagamet, and it took off immediately. According to a cost-benefit report published by 2 Harvard researchers, about 250,000 Americans were diagnosed with a new peptic ulcer every year back then. And in 1978, anywhere from 1.5 to 2 million American patients with peptic ulcers were treated with cimetidine. By the following year, 1979, the drug was sold in over 100 countries and earned over $400 million worldwide. In 1980, Tagamet got FDA approval as a duodenal ulcer maintenance therapy, which expanded their market even further since they weren't limited to acute ulcer treatment. By 1981, around 15 million people around the world had taken Tagamet, and it formally surpassed Valium to become the highest-earning drug in the United States. Cimetidine got so popular that it might have also lowered the rate of those ulcer surgeries. While the rate of these surgeries had been going down for the last decade anyway, there was a noticeably steep decline in 1978, the year after cimetidine came out in the US, from about 97,000 cases in 1977 down to 69,000 in 1978. This implied that some people were avoiding surgery because the drug allowed their ulcer to heal. Not everyone, but enough that it made an epidemiological difference. So, that answers part of this video's research question. Tagamet earned so much money because millions of people could use it, and of course, because it was the first drug in the anti-ulcer family and had a legal monopoly on the market before competitors popped up. This all led to 1986, when Tagamet became the first true blockbuster drug, a title defined by earning $1 billion in revenue in a year. But cimetidine wasn't perfect. Tagamet required 4 pills a day, and while the side effects were rare, it interacts with other drugs and can bind to androgen receptors, which, when used in high doses for a long time, can cause breast enlargement or sexual issues in men. So there was room for improvement. One of which came from the team of a researcher named David Jack. By coincidence, both Black and Jack were born in the same year in the same part of Fife, Scotland, and had almost parallel careers. Both were early adopters of rational drug design, and while Black developed beta-adrenergic receptor antagonists, Jack created beta receptor agonists, which culminated in asthma medicines like albuterol and salmeterol. Jack worked for Allen and Hansbury's, the research arm of the British company Glaxo. They were just starting to get into pharmaceutical development in the early 60s, and Jack was their guy in charge of picking which drugs to research. In April of 1972, the SKF scientists published their findings on the H2 receptor and their first full antagonist, burimamide. And at this point, they'd already submitted a patent for cimetidine, so they weren't giving away any secrets to potential competitors by publishing. But while the paper didn't give away the final product, it did show that histamine receptors were a clear target for ulcer meds. So Jack decided that Glaxo would pursue an H2 ulcer medicine of their own. Unfortunately, this is where the story gets kind of frustrating, because while there's so much publicly available information about cimetidine's development, I couldn't find much about Glaxo's process. To be fair, SKF had more to document since they were making something novel, while Glaxo was making a Me Too drug. What we do know is that, just like SKF, Glaxo had to decide whether to modify histamine's core ring, the side chain, or both. And they needed to do all that without violating SKF's patent. The patent covered drugs based on native histamine's central ring, imidazole, and a lot of scientists thought that this was the special sauce, that you couldn't build an H2 blocker without imidazole. But Glaxo built compounds around a different central ring, what's called furan, a 5-sided ring with an embedded oxygen atom. From there, they tried sticking different side chains on the furan ring until they arrived at this tail. According to an article the Glaxo scientists released in 1979, this new chemical that they called ranitidine was 4 times more potent than cimetidine, both in tissue samples and in dogs. After successful clinical trials, it was approved for the UK market in 1981 and for the US in 1983. Glaxo called it Zantac. But by the time Zantac came out, Tagamet had been approved for years and was growing like crazy, so Glaxo must have felt the pressure to get a chunk of that market. Now, Glaxo's biggest potential for sales was in the United States, but they didn't have a huge sales presence there yet, so they decided to partner with Hoffmann La Roche, who had more experience selling in the US. They were coming to the end of their patent protection on their big money-making drug, Valium, so they were looking for a new source of revenue. And thanks to this co-marketing deal, Glaxo's sales team tripled from 400 to 1,200 reps, and they were aggressive. Sales reps called detail men would call doctors and tell them how Zantac had the advantage of twice-a-day dosing compared to Tagamet's 4 a day, and it worked. Despite a 15 to 25% higher price than Tagamet, Zantac earned almost a quarter of America's H2 blocker market within its first year. But, in my opinion, the big advance for Zantac happened in 1986 when they got FDA approval for GERD, or gastroesophageal reflux disease. GERD involves stomach acid backsplashing into the esophagus, so this is another disease where tamping down acid secretion can help. And while doctors could prescribe cimetidine or ranitidine for GERD off-label anyway, getting FDA approval for a new indication comes with advertising benefits. In the United States, manufacturers are only allowed to advertise their drugs for the conditions they've been approved for by the FDA. So after this new approval, Glaxo was allowed to mention GERD in Zantac advertisements while Tagamet couldn't. And while I couldn't find exact numbers for the mid-80s, in the 2010s, anywhere from 1 in 6 to 1 in 4 North Americans had GERD. So this new approval expanded their total addressable market. And so, Zantac took the title of best-selling drug by annual global revenue in 1987 and stayed there for 10 years. And this is while the market is getting even more competitive. In 1986, a Japanese company called Yamanouchi came out with famotidine, which was more potent than the last 2 H2 antihistamines. Yamanouchi co-marketed with Merck to sell famotidine as Pepcid in the US. Within a year, it had carved out a respectable 8% of the H2 blocker market. According to the National Bureau of Economics Research, by the end of 1993, Zantac had over 55% market share, followed by Tagamet at 21%, Pepcid at 15%, and Eli Lilly's Axid at 9%. SmithKline and French weren't the big dog anymore. Speaking of which, by the early 90s, SKF had undergone some corporate mergers to become SmithKline or SKB, and they had a new challenge. Not only was there more competition in the H2 antagonist space now, but cimetidine's patent was set to expire in 1994, which meant they'd lose out on Tagamet sales to generic competitors. So SKB looked for a way to breathe some new life into their old blockbuster. And they did it with the same strategy that Zantac used. They got approval for a new indication. But this time, they chose heartburn. By the mid-1990s, the market for heartburn medicines was estimated to be worth $5 billion. So SKB took a lower dose version of cimetidine and applied for FDA approval as an over-the-counter heartburn treatment. They began clinical trials and got approval in August of 1995. Meanwhile, Glaxo, who had also undergone a merger to become Glaxo Wellcome, was going over-the-counter too. But while we're on the topic of mergers, there's a bit of an enemies to lovers twist in this story. In the year 2000, Glaxo Wellcome, the makers of Zantac, and SmithKline Beecham, the makers of Tagamet, merged into GlaxoSmithKline, or what we call GSK today. But the Zantac story doesn't end there. In 2019, independent scientists found that its active ingredient, ranitidine, degraded into a cancer-causing chemical called NDMA. In response, GSK and its co-marketers around the world recalled the prescription-only version of Zantac by October of 2019. The FDA formally advised a recall of ranitidine drugs in April of 2020. But the Zantac brand name lives on. GSK replaced ranitidine with famotidine, the active ingredient in Pepcid, and sold it as Zantac 360. Although, in a turn of events, the FDA approved a new reformulation of ranitidine in November of 2025, but the press releases have mainly lacked details. So, to recap, cimetidine came out in 1976 in the UK and became the best-selling drug in the world from 1981 through 1986. Ranitidine came out in 1981 and again became the best-selling drug in the world from 1987 through 1996. But even Zantac was no financial match for the gastric acid suppressor that came next, one that would work in a totally new and superior way. While SKF and Glaxo were working on their drugs, a Swedish group under the company Astra, one of the companies that went on to become AstraZeneca, started working on a medicine to block acid secretion too. In 1972, they found research from a French company who had recently published about some compounds that might reduce gastric acid secretion. But the company found that the compounds were too toxic during testing, so they canceled the project. The Astra scientists were pretty confident that they knew which part of the French group's compound was causing the toxicity, so they took the compound, kept the main ring, and started making new versions where they replaced the supposedly toxic part. By the next year, they got a compound that curbed acid secretion and wasn't as toxic, a benzimidazole codenamed H124. And they would have kept going with that compound, but a Hungarian company had already taken out a patent that covered their new drug as a tuberculosis treatment. But it didn't cover a metabolite of that compound. Basically the same thing, but with an oxygen bonded to that sulfur in the middle. Astra called it timoprazole, and it was more potent than anything they'd come up with before, so they pursued this one as their new lead. Unfortunately, during toxicology studies in 1974, they found that it led to enlarged thyroid glands and shrunken thymus glands. And that failure highlighted another problem. They didn't know how the drug actually reduced gastric acid secretion. But by chance, one of the Astra scientists happened to run into exactly the right guy to teach them. But for their meeting to make sense, we need to cover some physiology. First things first, you can tell that something is an acid because it gives off hydrogen atoms in water. And stomach acid is hydrochloric acid. It's the textbook example of a strong acid. The cells in your stomach that secrete hydrochloric acid are called parietal cells, sometimes called oxyntic cells. They've got those familiar receptors for histamine, gastrin, and acetylcholine that it gets from the vagus nerve, as well as receptors from prostaglandins, which should sound familiar from the aspirin video. But no matter what it's stimulated by, it all culminates in the final step, pumping out hydrogen ions. Just one problem. Chemicals tend to flow from areas of high concentration to low concentration, and there's a way higher concentration of hydrogen ions outside the cell, in the stomach cavity, than inside the cell. The hydrogen ions wouldn't flow out passively, so something had to be actively pumping them out. By the 1970s, researchers had found an enzyme in human stomachs that took in potassium ions and pumped out hydrogen ions. This became the leading explanation for how gastric acid was made. And since hydrogen ions are just protons, this enzyme became known as the proton pump. Alright, now we can get back to the story. It's the summer of 1977, and a chemist from Astra happened to meet a researcher named George Sachs, who was a big name in gastric acid physiology research. He was one of the guys leading the hypothesis that the proton pump was responsible for gastric acid. So the Astra chemist asked him whether this proton pump might explain the adverse reaction in the thyroid, and Sachs said that it might. They made the connection that maybe tomoprazole was an inhibitor of the proton pump itself. And it turned out it was. So Astra struck a deal with Sachs' lab in Alabama. Sachs' team would develop the first proton pump inhibitor, and Astra would sell it when it was ready. The team confirmed that tomoprazole was indeed an inhibitor of the proton pump enzyme. I'll link the paper in the description if you want to read the methods, but the research happened in 1978 and the results were published in Nature in 1981. They also found that the drug was a prodrug, meaning it needed to be metabolized before it turned into its active form. They also found that a similar drug, picoprazole, was much more potent. And while Astra didn't try to get this one approved, they did a proof of concept trial to show that it could lower gastric acid secretion. And in the background, they just kept optimizing the chemical structure, with the goal of getting the drug to accumulate around the proton pump for a more powerful effect. And in January of 1979, they created omeprazole. It didn't mess with iodine uptake, which is what caused the thyroid stuff, and was a potent inhibitor of gastric acid in rats. And just like its older siblings, Timo and Pico, omeprazole is a prodrug too. This is part of what makes it so effective. The drug turns into the active form when it's exposed to high acidity, like the area right outside a parietal cell where it's pumping out protons. Omeprazole is a weak base, and its structure is just right that it accumulates in the area before turning into its active form. It's a cool mechanism of action. Astra started phase 1 trials by 1982, but then in 1984, during phase 2 trials, long-term studies in rats made it seem like the drug might contribute to slow-growing tumors. So they paused trials, but it turned out to be a result of gastric acid inhibition in general, not the drug specifically. The same thing happened with long-term high-dose treatment with H2 blockers, as well as with a certain surgery. Trials started up again, and phase 3 trials went smoothly. These trials were enough for omeprazole to earn approval in Europe in 1988 for the treatment of ulcers, where Astra sold it as Losec, short for low secretion. In the United States, Astra decided to collaborate with Merck, who must have been stoked, because they had recently co-marketed Pepcid in the US, and now they were getting this new generation of ulcer meds to add to their portfolio. Merck helped Astra get everything ready for the US launch and got it approved by the FDA in 1989 as a prescription treatment for GERD, then later for duodenal ulcers and other indications. Astra sold it as Prilosec. It wasn't over the counter yet, but I couldn't find stock images of the old stuff, so this'll have to do. The important thing is that it's purple. In 1996, Prilosec took the top spot from Zantac and became the best-selling drug in the world. And by 1999, it peaked in sales, earning $5 billion that year. But more broadly, as a class of drugs, proton pump inhibitors were second only to statins in sales. In 1998, research published in the New England Journal of Medicine showed that omeprazole was more effective than ranitidine for patients with NSAID-induced ulcers. After 8 weeks, 80% of patients treated with omeprazole had healed, compared to 63% of patients treated with ranitidine. So hooray, money. But Prilosec had some downsides. Like, some people metabolized the drug more slowly than others, and some needed much higher doses to get any relief. There was a meaningful amount of variation between patients. Knowing that, Astra started a new research program to find a more bioavailable drug. So scientists spent the next 5 years tweaking omeprazole and testing it in rats, and they found one drug superior to omeprazole. And it was another version of omeprazole. See, molecules have three-dimensional shapes. And just like your hands, some molecules can have mirrored versions of themselves. It's a phenomenon called chirality. This is the case with omeprazole. There's a left-handed S version that mirrors a right-handed R version. Prilosec has both the S and the R versions. Astra's tests found that the S-handed version, what they not so creatively called S-omeprazole, was more bioavailable than original omeprazole. More of the medicine got from the mouth to the blood. And during clinical testing, it was better at reducing acid secretion and treating GERD. The company got approval for this one in 2001 and sold it as Nexium in the US. And it was controversial right away. At this point, Astra had become AstraZeneca, and they released Nexium right when Prilosec's patent was about to expire. And since it was basically the same molecule, Nexium just kind of seemed like a cash grab. Over the next few years, AstraZeneca did some head-to-head tests comparing omeprazole to esomeprazole, but for the most part, the new drug wasn't more effective than the old one at the same dosage. Like, in one study, the new drug healed 90% of erosive esophagitis cases after 8 weeks, compared to 87% from good old omeprazole. And it became this big controversy. The guy running Medicare Medicaid back then, Thomas Scully, basically said, "We're not covering a more expensive drug that doesn't work better than the old, cheaper one." In response, AstraZeneca doubled down and said, well, at least now patients and doctors have options. But then they did this big direct-to-consumer marketing campaign, plus jacked the price up for Prilosec, which made Nexium seem like the best alternative. It was classic evergreening. Either way, Nexium sales peaked in 2006 with $5.2 billion in revenue. Meanwhile, AstraZeneca still had this well-known brand with legacy Prilosec. So in 2003, after omeprazole went generic, they got Prilosec approved as an over-the-counter option for heartburn and partnered with Procter & Gamble to bring it to the market in the US. And just like Tagamet, that was a great strategy to get a little more revenue from their old intellectual property. Most people with frequent heartburn go to the pharmacy, not the doctor's office, and by their estimates, about 50 million Americans suffered from heartburn. Plus, that made it seem like Nexium was more powerful since it was prescription only still. Fast forward to 2012, and AstraZeneca partnered with Pfizer to market over-the-counter Nexium in the US. After that, it's a familiar story. Esomeprazole's patent expired in 2015, and generic competition drove the price down. But the rise of proton pump inhibitors solidified their spot as the go-to treatment for diseases like GERD, ulcers, and heartburn over the H2 blockers. To bring it to modern day, these days we know that ulcers can be caused by bacteria called H. pylori. It's a super common bacterium in human stomachs. So doctors usually treat ulcers with a combination of antibiotics plus a proton pump inhibitor. As we get into the takeaway section, I want to read you a snippet from a 1981 article in the New York Times headlined, "The Drug Business Sees a Golden Era Ahead." Quote, "This is the industry of the big breakthrough, where the introduction of one successful product can literally make a faltering company whole or pay for an expensive research program many times over. No one knows this better than SmithKline Corporation, whose stock multiplied 8-fold since 1975. The year before its ulcer remedy Tagamet was introduced." Later on, they quote a healthcare analyst who says, quote, "The name of the game continues to be getting the big product to come along and have an important effect on sales and earnings. All it takes is one hit every 3 to 5 years, and you can sustain all the risk." And that's the takeaway. The milestone of $1 billion in revenue isn't that meaningful. Valium hit hundreds of millions in revenue. But the $1 billion mark overlapped with a paradigm shift in pharma. These companies knew that investing in R&D was important, but the big payoff of a blockbuster like Tagamet made the risk of R&D more worth it. After all, SmithKline and French took 13 years of research to come out with Tagamet, but financially, all that time was worth the risk. Now, some factors have changed the pharma industry's risk tolerance since then. Like, this was at the beginning of the Reagan administration, which promised to deregulate lots of industries, and an overall aging population expanded the addressable market for certain drugs. But in modern day, a lot of risk is taken by biotech companies, and then pharma companies buy those biotechs. You can watch my video about Genentech for more on that. So while companies learned a long time ago that investing in research and development was worth it to bring new drugs to market, the introduction of cimetidine in the late 70s was the beginning of the big swings. Before you go, I care about the accuracy of these videos a ton, which is why I don't use AI to research or write scripts for me. I sometimes use AI on the same level as like Grammarly, but I found so many errors when trying to fact check Google's AI results or ChatGPT stuff that it's usually a waste of time for me to use AI during research. That said, I always link my sources down in the description for transparency. And sometimes it leads to some good follow-up reading. And for this video in particular, I also reached out to the archivist at Glaxo, who kindly let me use those archival clips of the SKF team. Likewise, all the other images and videos were either licensed through Getty Images or shot or made by a real person. Same thing with the intro music. It was created by an actual human. His name is Kevin MacLeod, and he is the GOAT of royalty-free music. So if you want to keep seeing videos like mine that are made by humans for humans, consider supporting me on Patreon. You get early access to my videos before they go public on YouTube, but most importantly, you're supporting the kind of non-AI slop that you want to see in the world. So if you're interested, head to patreon.com/corporus or scan the QR code on the screen. Thanks for watching. Take care.